Our next speaker is Dr. Alison Van Eenennaam. Alison is a member of the faculty in animal science at University of California, Davis. She received her bachelor's degree from University of Melbourne and her master's and PhD from UCD, and those were in genetics. So Alison is internationally recognized in her extension program at UC Davis, and that program focuses on DNA-based technologies to address applied problems of animal agriculture. She's a leader for the extension aspect of the USDA-funded integrated program for reducing bovine respiratory disease complex in beef and dairy cattle, the Coordinated Agricultural Project, or the CAP project, that is part of the sponsors for this symposium. So Alison has also served for the National Extension Representative of the U.S. National Beef Cattle Evaluation Consortium Industry Council, and she's been involved in validation of DNA marker tests for beef cattle since 2005. She's been involved in a lot of public education and uses a variety of media to keep public audiences well informed about important aspects, including cloning and genetically engineered animals. So please welcome Dr. Van Eenennaam.
Thanks very much, Laurel, and good afternoon. It's a pleasure to be here, and I thank all the preceding speakers because I think they've given me a very nice introduction to the topic I'm going to be discussing here today. And it's nice to follow Donna because he's got a more difficult accent to understand than myself. So I will try to go through and really represent the efforts of a large multi-institutional team that's shown on this slide here, which is the BRD CAP. And I'm going to just discuss briefly what the BRD CAP is, and then move into the area of genomics that was so nicely introduced by the preceding speaker, and discuss some of the research objectives of the BRD CAP, the progress we've made to date, and then some of the translational considerations, which as an extension specialist is my area in this interest in this particular area. So the BRD CAP, for those of you that are not familiar with it, is a coordinated agricultural project. It's a large multi-institutional 5-year grant that is composed of the researchers that are shown on the slide here. And Dr. Jim Womack at Texas A&M is the project director of this particular project. And a lot of the data I'm going to be presenting here today is from the research team that's led by Dr. Holly Nybergs from Washington State University, who's present in the audience here today, and members of that research team. I'll also just talk about the fact that Milt Thomas at Colorado State University is in charge of the educational aspects of this project, and I'm in charge of the extension attributes of this project. And basically what this project is trying to do is bring genomics, that is the study of the entire genome, to the problem of BRD. And, you know, genomics is particularly well suited to problems where we don't have very good phenotypes. And part of the background and rationale here is that, as we've discussed this morning, and I think Mike Engler did a really nice job talking about the fact that despite all of our management and vaccines, we're actually seeing a slight uptick in mortality rates each year. And we've been researching this disease since the 1800s, and we're actually still going in the wrong direction. And so there was a feel that there was a need to develop new approaches to tackle BRD, and is genomics an approach to do that? And genomics is particularly well suited to traits that are difficult or expensive to measure. And I would put to you that disease resistance or susceptibility is one of those traits. This is a very expensive approach to testing whether or not an animal is susceptible. They're very well suited to traits that can't be measured until after selection decisions have been made. That's very typical for this particular characteristic, where the animals are often in the feedlot before they display this particular phenotype, and they're not currently selected for because it's just very difficult to get measurements on these. And as we know, often animals that are going through the beef supply chain, the phenotypes of those animals in the feedlot do not get transferred back to the breeders that are making selection decisions, and so we have no selection criteria upon which to base selection decisions. So the presence of genetic variation in resistance to disease, which we know exists, coupled, I think, with some of the talks from Dr. Apley this morning with increased consumer resistance to the use of antimicrobials, makes a genetic solution to this problem a very attractive proposition, and also the resistance that we're seeing in some of the antimicrobials. And we know that in other industries with well-structured breeding programs, in particular the dairy industry and the poultry industry, they have been able to develop selection criteria against disease traits, for example, somatic cell count in the case of dairy cattle, where they've been able to make selection decisions that decreases the susceptibility to those diseases, whilst also making genetic progress in the other traits of importance to the breeding objective that bring economic revenue back to the producer. So the long-term goal of the BRD CAP then is to reduce the incidence of BRD in both beef and dairy, and I think that's an important component, by capitalising on recent advances in genomics to enable novel genetic approaches to select for cattle that are less susceptible to disease. This is a very large project and it has multiple different research components, and today I'm really just going to focus on one research objective, which is to identify genomic regions associated with BRD resistance, susceptibility in beef and dairy cattle. So I want to talk a little bit about what the genomics revolution has delivered to date, and basically with the first sequencing of the bovine genome, which was less than a decade ago, we started to find out that we were able to find a lot of genetic variation, or what's called single nucleotide polymorphism, or SNP markers in the genome, that enabled us to basically have a marker that would tell us give us a genetic polymorphism, and what we were able to do was develop multiple polymorphisms, or in the case of the 50,000 SNP chip, 50,000 little markers that are spread evenly through the 3 billion base pairs that make up the bovine genome, that basically give us a little flag or a handle to say what that particular animal has at that location, be it a T or a C. And what we need to do to develop DNA tests for BRD susceptibility or resistance is to develop these large training databases where we have a phenotype, in this case disease resistance. In the case of the dairy industry, let's just talk about milk production, because this is an example where they've made very great progress in this regard, because they had very large data sets of milk records on very highly used AI bulls. And they were able to basically genotype those animals, those high accuracy AI bulls for milk production, and estimate the effect of the 50,000 markers on milk production. And so basically it's kind of like getting a genetic merit estimate for the 50,000 markers that are spread throughout the genome. And once you've used this large training or discovery population to estimate the effect of each of the genetic markers with your trait of interest, you can then go in and develop what's called a prediction equation that allows you to then take a DNA sample from an animal for which you have no phenotypic information, be it milk production in dairy industry's case, be it BRD susceptibility in this particular example, and you can predict the genetic merit or the breeding value of that animal using this technology. And it's really the power of the sequencing the genome that's given us these markers that's enabled us to start using this information to make better selection decisions. And in the case of the dairy industry, it's probably been one of the most rapidly adopted technologies, I think, in animal breeding, where in 2009 we had the release of the 50,000K SNP chip, as it's called, which is now being used routinely in the breeding industry throughout the world to more accurately predict the genetic merit of dairy bulls, young dairy bulls, and then select the ones that are going to go into AI programs. And so the concept is, how can we develop a large training population of animals that have disease resistance phenotypes for BRD that would enable us to develop this training population that then would give us the prediction equation that would allow us to go and predict the genetic merit of animals in the field. And since the 50K chip came out, we now have the power of the 700,000 SNP chip, which gives us even more markers spread throughout the genome, and even better ability to define each of the 700,000 little segments of the genome and have a look at the effect of that particular little segment of the genome on our trait of interest. And so it's given us this ability to really dissect the host variation and identify possible genes contributing to this variation using what's called genome-wide association studies. But the disadvantage or the problem is that in order to have enough power to tell the test the effect of 700,000 little segments of the genome, you need to have these very large training populations. In the case of the dairy industry, they were kind of lucky because they had very nice records that were sitting in the Dairy Herd Improvement Association, and they had semen straws sitting in a tank that gave them the ability to extract DNA, run the genetic test, and then they could do their prediction equations. We don't have a repository of DNA and phenotypes for BRD resistant susceptibility sitting in a repository like the dairy industry did. And so part of the rationale behind the BRD CAP was to develop these large training populations for both beef and dairy cattle in order to give us the ability we need to do this training and work out which segments of the genome are associated with susceptibility or resistance to BRD. And to get these large training population, this is not an experiment that can be done in a university feedlot. We need thousands of individuals, and that basically requires the use of field data. And so one of the tenets of the BRD CAP project is we've been collaborating with industry, both dairy and beef, to work with large enterprises that in the field to try to develop these training populations. The problem is, as been discussed in multiple different talks this morning, is field data in this regard is very noisy. The diagnosis of whether an animal is sick or well is imprecise and can be difficult to determine whether infection of an actual individual has occurred. Was it actually exposed to disease and resistant, or did it never get exposed to some type of a pathogenic challenge? And it's often unclear whether apparently healthy individuals have been exposed to infection. And what this does is it has the tendency to give you increased noise, because you might misclassify an animal as a case when in fact it's not sick, and you might misclassify an animal as not sick when in actual fact it is a case. And that basically lowers the heritability because you have a lot more environment, a lot more noise in the background. And so one of the really important features of these data sets was try to develop criteria to select case and controls that was very constrained. And so I think the traditional methods, as has been discussed this morning, are basically the DART system and rectal temperature, and temperatures can lie to you, and a lot of these are subjective in nature, and so they can be confounded by the diligence and astuteness of those checking for the symptoms. And I think that perhaps all of us could have a look at this animal, and I don't think any of us would probably have a hard time determining whether or not this might be a case or a control. And I know that he's a case because we actually intentionally infected this particular animal, and his pen mate round the corner isn't looking much better either. But what we tried to do was have a look to take the fact that it is very hard to get these case definitions to be accurate, and tried to standardize it using the scoring system that was discussed by Sheila McGurk this morning. So I'm going to discuss the field data sets that have been and are being collected by the BRD CAP, and discuss in some detail the dairy population where collection has been completed. And so basically what we've done is collected 2,000 case controls, so 1,000 case and 1,000 controls from dairy calves on a dairy calf ranch in California, and this is the same data set that Sharif Ali was speaking about in his talk before the break. And we also have a similar data set getting collected on feedlots in Colorado and Washington. And then we have a confirmatory or a validation data set getting collected on an additional 763 dairy cattle in New Mexico and 1,000 or so beef cattle in Nebraska that will be used to validate the loci, because we want to make sure that the loci that we're finding being associated with susceptibility can be repeated across different populations. And that's been one of the problems with some of the genetic tests that have come out until this point, is they haven't always validated well. And it's a really important thing to make sure that we're actually still getting accurate prediction of genetic merit of animals that are susceptible or validated before we go to the field with these tests. And so all of these animals are going to be genotyped on the high-density SNP chip, and the pathogens will be characterized using bacteria and virology. And I will talk about that, but that kind of gives us the opportunity to look at genotype by pathogen interactions. So, as was discussed by Dr. Ali earlier, this is a collaborative effort. There was a lot of involvement of the vet school from UC Davis, Terry Leyenbauer, who's in the audience here, and a veterinary intern, Jessica Davis, who went through and screened these animals looking for animals that were cases. And basically we used Dr. Sheila McGurk's scoring system that Sheila's nicely introduced, and so I won't need to spend time going through what that was. And we said that a case was an animal with a 5 or greater using her scoring system. And then we collected samples for DNA collection, and then deep pharyngeal nasal swabs for bacteriology, and deep pharyngeal nasal swabs and a nasal swab for virology. So Sheila's gone through this. These are some of our case animals in the California study. And here's some pictures of blood collection, nasal swab collection, deep pharyngeal swab collection. And as was mentioned by Dr. Ali, for the bacteriology, we tried to get a deep pharyngeal swab to minimize just environmental organisms that were present in the nasal passages. And the controls were scored in exactly the same way, and ideally what we tried to do is once we enrolled a case, we tried to enroll a control that was in the same general environment, ideally the next hutch, that would likely have been exposed to the same pathogens or microbes that the case had, and that wasn't sick. And we collected samples from those animals. And what we were trying to do was objectify our selection of cases and controls to have the minimum amount of subjective information and try to dampen down that noise such that we could increase the amount that's due to genetics, or in other words, try to get a better heritability, increase the heritability of that. And so the preliminary results of the dairy study, so it's estimated that over 200,000 calves at this large calf ranch were evaluated, and there was a total of 1,000 cases and controls picked out there. And there was a positive association between a McGurk score of greater than 5 and all of the positive cultures for BRSV, Mannheimia hemolytica, Pasteurella multocida, H. somni and Mycoplasma, which gave us some level of comfort, and I think that the scoring system is doing a good job at picking out sick animals. Interestingly, all of them were DV negative. But then the important thing comes now from the GWAS studies, and these GWAS studies were done in 3 different labs. So Dr. Holly Nyberg's at Washington State University, Chris Seabury at Texas A&M, and Jerry Taylor at the University of Missouri took the same data sets and did their favorite analyses. You know how quantitative geneticists are. They all have their favorite program. And I think very encouragingly, the heritability estimates we were getting was 20%, and that's higher than some of the estimates that have been found in previous studies that Donna alluded to. We believe it's because we've really tried to get a good objective definition of cases and control and minimize that environmental variation and really pull up that genetic signal. And then the SNP effects at this time are explaining about 20% of the variation in BRD incidence and 17 to 20% of the variation in clinical signs. And so I think that's very positive data to date. The heritability is very encouraging, and as was shown by Donna, if you can get heritabilities up there, it really gives you the opportunity to make some genetic progress. If we look at the preliminary results, there's 3 areas that all 3 labs came up with, 3 chromosomal areas that were very positively associated with BRD susceptibility, and there are additional areas on other chromosomes where 2 of the 3 labs came up with the same area. And so these are a preliminary analysis, and there's now subsequent work to try to hone in on or develop a smaller region that's associated with BRD. And ideally what we would like to do is actually get down to what's called the causative SNP, or the actual DNA variant that's resulting in that difference in BRD susceptibility, because the actual causative variant, rather than a marker that's just close by, is going to be the best thing to use for some sort of a diagnostic test that will hopefully be more likely to work across different breeds. And so there's a lot of effort now to do fine mapping and use the fact that we can now basically genome sequence entire bulls for a relatively small amount of money, which is mind-blowing given it's only been 5 or so years since we sequenced the first one, to try to get these more predictive mutations. A similar study has been undertaken in beef cattle. We did choose to focus on Bos taurus cattle. There's quite a lot of genetic difference between Bos taurus and Indicus, and given this is a fairly ambitious project, we tried to stay within Bos taurus to maximize our chances of success. And there is sample collection that has been completed in Colorado, where 1,000 samples have been collected, 500 cases and 500 controls, and there's ongoing sample collection at the current time in Washington State on a feedlot there, and there has been some preliminary analysis of the beef data, and again, very encouragingly, the heritability estimates were 17.7% if you just looked at a case control or a 0-1. If you actually use numerical values of the McGurk system, so 1 through 12, you could actually increase that heritability up to 30%. And that's a lot higher than the estimates that have been presented by Donna and it found in other studies. And again, we believe that this is because we've managed to try to minimize the environmental differences that would otherwise cloud the heritability or the genetic causes there. It should be noted that these GWAS studies, although they really are the bread and butter of the BRD CAP, are just one component of an approach to look for genetic variation and to try to identify the genetic regions that are associated with BRD susceptibility. And there are other researchers that are working on different approaches. There's researchers at Texas A&M, Lauren Scow and Scott Dindo, looking at copy number variation, which has been seen to be very important in disease traits. Laurel Gershwin, the moderator of this session, is working with Jerry Taylor at Missouri to challenge animals and have a look which genes respond to different pathogens and bacterial and viral pathogens. Holly Nybergs is looking at pathway analysis to try to identify pathways of importance, and Cassie Tucker at UC Davis is looking at behavioural studies. And it's envisioned that jointly the data from all of these different approaches will result in information that will be required to enable the much needed development of DNA tests for the selection of animals that are less susceptible to BRD. And then I guess the thing to me is, okay, that's nice. The researchers have got their markers. Now what do we do? How do we translate this to industry? And so, of course, first we have to validate that the SNPs and the areas that are lighting up as being associated with disease susceptibility or resistance are in fact found in other populations. And then the important thing to do is then to start incorporating that information into your breeding values or your breeding objectives, your selection index. And Kurt Van Tassel is working on that, and he's basically developing genomic breeding values for Holstein sires that are less susceptible to BRDC. And ultimately, we'll need to incorporate that trait into the selection index that's used in dairy cattle production. And dairy has it a little bit easier just because of the prevalence of the Holstein breed. And so the appropriate selection emphasis that should go on a trait like BRD will really depend on its relative economic importance, considering the other traits that make the producer money. And I just want to have a look at the dairy cattle economic selection index as it exists at the moment. And I know a lot of you are beef veterinarians, but it's kind of interesting to me, if you look at the traits that are currently in the breeding objectives of the dairy industry, there's actually zero emphasis on milk. And I guess that's a little counterintuitive if you think, I kind of thought that's how dairy producers make their money. But it's because there's positive correlations with milk fat and milk protein that have positive things here. But I want you to also pay attention to the fact that there's a relative selection emphasis of about 10% here on mastitis resistance. So they've done the economic analysis to determine that that's about the right emphasis to put to select against mastitis relative to all of the other competing selection criteria. Because if you put a lot of emphasis on, say, milk fat, 100%, then you'll get drift according to the genetic correlation with other traits. And so it's important to have this selection index optimally weighted for the economic production system that you're working in. And to Donna's point, here's daughter pregnancy or fertility with about an 11% weighting, and as you saw, what that was able to do was turn around that genetic trend in fertility and start going in a positive direction. Dairy have it a little easier than the beef industry, and there's a couple of reasons for that. And it's a bit of a more challenging situation in the beef industry. One of the things we've found is that if you use the 50K prediction equations in Angus, it doesn't predict well in another breed, Hereford, for example. I think that was unexpected when we first started this project, or at least not this project, but when we first started with genomic selection. And so it was envisioned that by using the higher density chip, that is the 770K chip, where we'd have a lot more markers that would hopefully get us closer to the actual causative SNP or gene that's associated with susceptibility, that we might be able to get prediction equations that would be more translatable across breeds. And it's not yet known if that will in fact be the case, but there's also, as we're going along, technology is advancing and we're starting to get whole genome sequences on bulls. Actually, thousands of bulls are now been whole genome sequenced, and there's a real hope that we're going to be able to use that information to identify actual causative SNPs, as I mentioned earlier, and that's much more likely to be translatable across breeds because you've actually got the causative mutation rather than a marker that's linked to it that may differ in phase between one breed and another. So conceptually, in the beef industry, genomic information shouldn't require any fundamental changes to the development of a selection index, and the selection for genomically enabled traits like BRD resistance will have to be weighted by its effect on profitability, but there's a need to determine what is the relative economic value of BRD resistance, for example, relative to the other traits that make the producer money, weeding weight or growth rates, for example. There's a paper was presented at BIF by Shannon Nybergs from Washington State, which estimates that the feedlot industry could gain between 8 and 16 million per year through the implementation of selection for cattle that are less susceptible to BRDC. But until this point, the beef industry has not made a lot of use of economic selection indexes. I still see a lot of producers maybe selecting on one EPD, maybe calving ease, if that. And so I think that although there's undoubtedly huge value associated with a selection against BRD susceptibility, it needs to be incentivized in some way, especially for producers that are making selection decisions who are not necessarily seeing the value of that selection decision because they're selling their calves at the sale yard, for example. And Mike McNeil and I did a study where we estimated the economic value of selecting against BRD or selecting for BRD resistance is actually valued about 4 times higher than selection for growth traits. And so it should be given a very high emphasis in a rational breeding objective for the entire beef industry. But with the segmentation of the beef industry, there's a little bit of a, I guess, difficulty getting the breeders and cow-calf operators to put that much emphasis on a trait like that if they don't necessarily see the returns associated with that. And that gets back to some of the earlier talks about the difficulty of trying to get producers to implement both genetic and management approaches that benefit downstream sectors when they don't necessarily get the return to that. So conceptually, I think that we could do this. It's a matter of appropriately putting it in the selection indexes and rewarding producers for taking that particular selection criteria into their selection indexes. Because if they put selection against BRD against BRD susceptibility, it will have an effect on the progress they can make in other traits that make them money in that selection index. So in summary, the BRD CAP has developed these large case control data sets that are really going to be required to develop genomic breeding values for BRD susceptibility, and the heritability estimates are moderate, not low, but moderate, which is really encouraging. The GWAS analyses look promising. We need to validate that, in fact, the genomic regions we're finding validate across different populations and hopefully narrow down the areas to, ideally, causative SNPs. The economic value needs to be appropriately weighted in selection indexes and then put into those selection indexes. And it's likely that the translation to the dairy industry is going to be a little bit of an easier discussion than it is translating it to the beef industry, just because of the different industry structures and breeding objectives in those two different industries. So with that, I finish with a slide of the BRD CAP, and I encourage you to look at the website if you're interested in some of the information that I've discussed here today, and I thank you for your time.